Hello, my name is Diana Suarez, and today we will be discussing mechanisms of sound transduction in the mammalian Cochlea. Our objectives for the day, we're going to go over cell structure and mechanotransduction, tuning mechanisms, synaptic transmission, and we're going to go over a study overview and experimental findings, and then we will end with our conclusions and applications. So the cochlea's hair cells, which provide the cellular basis of auditory signal transduction, are the subject of this section. So in particular, we look how mechanical inputs are transformed into electrical impulses at the stereocellular level. The two different kinds of hair cells found in the mammalian cochlea are called inner and outer hair cells, and they are organized along the basilar membrane. What we can see here on the image. And despite having structural similarities when viewed under a light microscope, their functions and biophysical characteristics are very different. As the real sensory receptors in the inner hair cells, they are primarily responsible for converting mechanical information into electrical chemical signals that are transmitted by the auditory nerve in the central nervous system. So if you did take a look at the second arrow here, the primary source of afferent auditory information, these cells are in the charge of encoding the timing intensity of sound. On the other hand, outer hair cells serve as cochlear amplifiers. They improve the mechanical motion of the basilar membrane through somatic electromotility instead of relying on sensory data, particularly for low intensity stimuli, and this amplification for essential for enhancing hearing sensitivity and frequency selectivity. The stereocellular bundle is a complex of array actin field projections on the apical surface of the hair cells. Is the core structure for mechanotransduction in both cell types. These bundles are connected by tips that are crucial for channel gating, and the sound is induced vibrations displaced in the basilar membrane. The clitoral membrane causes the bundles to deflect, increasing tension in the tip links. This tension is transmitted to mechanotransduction channels located at the lower insertion of the tip link on the shorter stereocell. Tip links in tercilia function as macular springs that gate what are known as MET channels, and response to mechanical force. Opening these channels allows cautions to enter, generating a receptor potential and driving neurotransmitter releases. Calcium-dependent adaptation is crucial for MET channel behavior, allowing hair cells to adjust channel gating in milliseconds. This dual mechanism exemplifies the sensitivity and speed of auditory transduction, preserving sound intensity and timing with high fidelity. Calcium plays a recurring role in the MET adaptation and synaptic release. Tuning hair cells, topic organization, or the spiral mapping of sound frequency throughout the length of the basilar membrane is one of the cochlea's distinguishing characteristics. Low frequency sounds are felt at the apex of the cochlea, where the basilar membrane widens and becomes more flexible, whereas high frequency sounds are perceived at the base when the membrane is stiff and tight. Although frequency selectivity is made by the mechanical gradient, the sharp tuning and sensitivity seen in vivo cannot explained by it alone. The outer hair cells' electromotor characteristics are the source of such improvement. Prestin is a special membrane motor protein found in outer hair cells. Permits a somatic electromotility, which is a quick voltage-dependent change in the length of the whole cell. So this enables the outer hair cells to feed mechanical energy back to the basilar membrane by contracting, expanding, in response to receptor potentials. Let's look at the image closer up. If you do see, the outer hair cells have a sharp tuning curve contributing to fine frequency discrimination. Their voltage-dependent nonlinear capacity reflects Preston activity. The feedback location, you do see there, is with the outer hair cells in different regions having distinct properties. Also, the cochlear tuning If you do see, mechanism integrates passive biomechanics, active feedback, and instructive electrical renaissance. So now let's look at the inner hair cell's synaptic transmission mechanism, which has been modified for high fidelity auditory communication. The special structure of inner hair cells, which are ribbon synapses, allows them to release neurotransmitters a sustained and temporally accurate manner. So they're making them more merely like passive transducers. And there's dozens of synaptic vesicles that are attached to the ribbon in a dense electron-dense structure. So it's close to the presynaptic membrane. So for neurotransmitters to be released continuously in quickly response to auditory events, this setup is necessary. And let's look at the image to understand it a little bit more. So like you do see, each ribbon is closely associated with a voltage gated calcium channel, which form kind of like calcium microdomains. So they're extremely localized regions where calcium concentration is very high. These microdomains form immediately adjacent to vesicle release sites, reducing diffusion time and ensuring vesicle fusions occur almost immediately upon calcium entry. So this setup underlies a key phenomenon known as phase locking, where auditory nerve fibers fire action potentials in precise, they're synchronized with the phase of the incoming sound wave. So this temporal precision is critical for encoding sound frequency and rhythm, especially at low frequencies. Now, if you look at the image here, we're seeing more of where the calcium is at, and it is passing through. You do see the arrows. So this is more of a precise chemical synapse in the nervous system. So in the most recent investigation, researchers tried to aim to test the hypothesis that fidelity of synaptic transmission in the inner hair cells depend on both spatial proximity between calcium channels and release sites. So the buffering capacity of the intracellular environment is what it's connected to. So to do this, what they employed is a whole cell patch clamp electrophysiology in the inner hair cells from rodent cochleas. This technique allows precise measurement of iconic currents and synaptic releases by gaining electrical access to the inside of a single hair cell. And here, this following image on this next slide, we're going to see what goes with what went with the study. Continuing from the previous slide to this image, take a look at the images from 4A through 4C. They apply different intracellular calcium buffers. So either EGTA or BAPTA help to manipulate how far calcium could diffuse before being sequestered. So EGTA is a slow buffer allowing calcium to travel further, while BAPTA binds the calcium quickly and limits its spread. So this allows them to model how the spatial distance between calcium influx and vesicle fusion influences the speed and reliability of neurotransmission. So they can then measure exocytosis by tracking changes in the membrane capacitance, and then increase when vesicles fuse with the cell membrane, and by recording the excitatory post-synaptic currents from afferent neurons that form synapses with the inner hair cell. So this setup allows the researchers to link calcium dynamics directly to synaptic output, giving them insight to how tightly coupled the vesicle release machinery is to the source of calcium entry. So the results of the experiment were very striking and very informative. So the most important finding was, first of all, that effective vesicle release required calcium sources to be within roughly 20 nanometers of the vesicle fusion machinery. So this ends up being an extremely tight spatial coupling between a couple of channels, and they docked pneumatic vesicles. So this ends up going into the same image as before, and we look at the rest of the letters. So this is supported from figure D through F, which shows how the application of different calcium buffers affect the somatic responses. So when the fast buffer, which was the BAPTA, was used, it significantly reduced the EPSC amplitude. So that increased the latency of the somatic responses. This implies that the vesicle fusion is extremely sensitive to local calcium levels and cannot function properly when the calcium signal is disturbed even slightly. So notice now in panel D, the increasing BAPTA concentration progressively reduced the release of the probability. So this supports the idea that a nanomain model, whereas calcium must rise sharply and briefly to be in a confined space. So it's essential for the phase-locked and high-fidelity neurotransmission. So in the opposite way, EGTA had a minimal effect on the release probability and the timing. So this ends up suggesting that calcium did not need to travel far to reach its targets, but these findings started to demonstrate that the inner hair cells are optimized for speed and precision through spatial organization. So most importantly, this configuration also helps explain how the auditory system can encode sound waves in a sub-millisecond resolution, which is necessary when detecting sound localization cues and speech rhythms for any mammal. So now going into in our summary, the study did conclude that synaptic fidelity in the inner hair cells depends on an exceptionally tight spatial organization of what are calcium channels and vesicle docking sites. So this spatial precision allows for rapid and reliable vesicle fusion in response to even minimal depolarization. If you do look at the image, it is labeled A through B. They present a model of this specific architect. Here we see how the synaptic ribbon serves as an organization scaffold anchoring vesicles very close to the clusters of the channels. So the figure also depicts how calcium microdomains form directly on these docking sites, supporting synchronized neurotransmitter release across many synapses in a single hair cell. So the organization is not really arbitrary. It is actually a key functional adaptation. So this supports the temporal accuracy of auditory signaling, which is very important. We need to have fast responses to sound. So such fidelity allows auditory nerve fibers to fire in synchrony with sound wave cycles, preserving information about pitch, timing, and rhythm. So clinically, these are more broad implications. For example, any disruption to this coupling, whether through genetic mutations or affecting the calcium channel function or synaptic architecture, it could lead to auditory processing defects or even deafness. These findings not only improve our understanding of the basic sensory physiology, but it may also guide future therapeutic strategies for treating sensorineural hearing loss. So in the whole context of this understanding, this study exemplifies the molecular architecture that shapes neural coding strategies and sensory systems. So it's an elegant interplay between structure and function. So thank you for joining me today. These are my references.